So next up we have Dr. Bill Andrews. Bill is a postdoctoral research fellow at John Parnavella's lab in the Department of Cell and Development Biology at UCL. He obtained a bachelor's degree in biochemistry in 1990 and has been a research fellow. Maybe you didn't. Has been a research fellow at UCL since 1995. Today he'll be discussing the molecular mechanisms involved in cortical interneuron migration.
Well, first of all, I'd thank you for giving me the opportunity to give a talk today. This is going to be, because it's such a short period of time, I thought it's more to give you an idea of some of the ideas of what we work on rather than sort of real sort of data that we've got, although there is some in my talk. Okay, so as so on this part of the slide, these are some drawings, probably by Ramon Cal, who got a Nobel Prize, probably at the end of the 18th century, and these are different staining mechanisms that you use for staining neurons. And as you can see, that the adult cerebral cortex is a highly organized structure, and it's composed of multiple neuronal types. But basically, there are only two major types in the adult nervous system. You get the inhibitory GABAergic neurons, which account for 20–25% of these neurons, and these are born in the medial ganglion gametes, or MG, and they migrate tangentially into the dorsal cortex. The vast majority of neurons in the cerebral cortex are the excitatory glutamergic neurons, and they arise from radioglial cells lining the surface of the lateral ventricle, and these migrate radially before they arrive at the position in the cortex. And as we know, any perturbations in either the connections, the number of neurons, have resulted in a wide variety of neurological disorders, such as epilepsy, autism, and schizophrenia. This is just a quick schematic showing that interneurons are actually comprised of a large family of neurons, which can be distinguished morphologically by their shape as well as different chemical properties. So these neurons, they're born in the ganglion gametes, in which there's medial, caudal, and lateral. Most of the ones that are born in the lateral ganglion gametes project to the olfactory bulb, and they do that throughout adult life, which is one of the natural sources of stem cells that you get within the adult brain. Whereas the vast majority of cortical neurones are derived from medial ganglion gametes, and you also get some from the CGE. So, as you can probably see in this schematic, interneurons are generated in the medial ganglion gametes actually have to migrate to a long distance before they arrive at their position in the cortex. And they, during early stages of development, they form this structure called the striatum, which will become more evident later on, but they migrate in three streams within the cortex. Once they get into the cortex, they then migrate radially to enter the cortical plate, where they make connections with pyramidal neurons. Let me get this to work. So this is a quite cool video, if it does work, of a flat mount of the cortex of mice in which all the interneurons were labeled with GFP, and this will just show that, that these neurons migrate all over the place, completely cover the cerebral cortex. So I think that's quite cool. So in our lab, what we're interested in is what the factors that regulate and control this migration. We're predominantly interested in chemoattractive and chemorepulsive molecules. So as you can see from this sort of schematic cartoon, SNPs are chemorepulsive molecules, as are the semaphorins, SEMA3As and SEMA3Fs. So what we know is that interneurons are born in this region. They express roboreceptors, which are receptors for the SNPs. They also express neuropilins, which are receptors for the semaphorins. And as you can see, they're being forced away from this region. They tend to avoid this striatal region because they express the receptors, and then they get moved towards the cortex, where they're attracted by a whole range of factors such as SDF and neuroregulates. So we've been interested in sort of studying these different chemoattractive and repulsive molecules. And one of the ones we've done a lot of work on in our lab is the ROBO1 receptor. We work on, this is a family of 4 different receptors, and there's 3 different slit ligands, but this is the only one that showed any interesting phenotypes for us. So what we discovered was that this receptor is quite strongly expressed in the protective zones where interneurons are generated, and along the migratory routes that they sort of take. And they're also expressed in neuronal progenitors as stained with PH3, which is a phosphone, which is a mitotic marker. And what we noticed when we looked at this mouse, staining it with galbini, which is an interneuronal marker, as you see in F, which is a wild type mouse, the staining is quite weak, but in G you notice the staining is a lot stronger, suggesting there may be more interneurons. But what we notice in this region here, which is striatum, is that in the wild type mice, there's very few interneurons there because they're forced away by the action of some of these chemorepulsive molecules. But in the robo knockout mice, these interneurons seem to enter this region. So we decided to go away and make another knockout mice for robo1, and this time we mated it with a transgenic mouse where all the interneurons express GFP. And again, you can see that if you look in the cortex of these mice, there's a lot more interneurons in the cortex of this mouse. And also you notice that there seem to be maybe more in the striatal region. This is a bit interesting because this region doesn't express SLIC, which is the normal ligand to which roboreceptors respond. So we were wondering, since we know this region expresses semaphore 3A, sema3F, we wondered whether robo may actually participate in this other signaling pathway. So in order to do this, we first did some carried out sort of the chemo microtree assay. So we used this thing called a Boyden chamber. So in this sort of assay, you can actually put different chemoattractive molecules at the bottom, whether attractive or repulsive, and then they're separated by a semi-permeable membrane. And then you can put your cells on the top and see whether they're attractive or repulsed by whatever molecules you have underneath. So when we take either heterozygote mice for robo1 or knockout mice and treat them with just normal conditioned medium, we don't see any differences in migratory ability, so suggesting that the knockout ones migrate the same as wild type. But when we gave them semaphore 3A, we noticed that in the heterozygote mice there was less migration compared to conditioned medium, suggesting they're responding to this factor, which we already knew. But what we find is in the robot knockout mice, they don't seem to respond to semaphore 3A anymore, which is quite interesting. And they also did the same with sema3F. So we're wondering why they don't respond to this mechanism. So another way of doing this assay, the previous one was really quite quantitative, but another way of doing it is if we take bits of MGX plants and culture them and then give them either just conditioned medium or conditioned medium that contains sema3F, and look at the ability of the interneurons to migrate up the X-plant, you'll see that in either robo1 heterozygote mice or knockout mice, if we give them normal conditioned medium, they migrate out equally as well. But then when we give them a source of sema3A, you notice that in the wild type, they're not able to migrate out. But in the robo1 knockout, they still can. And we saw the same results with both sema3A and sema3A. So it looked like for some reason these neurons from these knockout mice aren't responding to these different factors. And we sort of went away and had a look, as we know that the normal ligand, we know the normal receptor for these receptors are the neuropilins of the plexin. So when we looked at either wild type mice or knockout mice, we looked at the level of the receptors in these. We noticed that in the robo knockout, there seemed to be less than neuropilin 1 and plexin A1, but not some of the other plexins. When we did the Western blots for these, again we noticed that there seemed to be reduced levels of the neuropilin 1 and plexin A1 receptors, which suggesting that since they express less receptors, they're not able to respond to the ligands for these molecules. So the third part of this, we just say that it looks like these interneurons seem to express ROB1 receptor, and we find an increased number of these of interneurons in the striatum and cortex. This is partly due to increased proliferation in these mice, but also it appears to be the ones in the striatum because they're not responsive to these semaphore molecules. And this is because they, for some reason, these robo mice, they seem to have less receptors. So if you don't have receptors, you can't respond to ligand. So one of the other things we're interested in about is what determines the choice of the migratory path of interneurons in the cortex. So actually they seem to go into different streams. So these are interneurons being labelled with di, and you notice that these interneurons seem to be heading towards different streams within the cortex. So we were wondering what factors were responsible for this. So in order to look at this, if you look early enough, there's only two interneuron streams that go into the cortex. There's one in the premotor layer and the intermediate zone. So in order to look at this, what we did is we did some laser dissection of cutting out these green interneurons from these two different streams, carried out a microarray, and see what genes were different between these. Interestingly, we found very few transcription factors, but we did notice a large range of different receptors. And one of the groups of receptors we noticed that showed quite a bit of differential expression were some of the cadherin molecules. And these have been shown to play a role in cell-cell interactions and in neuronal development. But more clinically relevant is that two of the ones that we found, like cadherin 8 and cadherin 13, have been implicated in autism and learning difficulties, and cadherin 13 has been shown to play a role in ADHD and bipolar disorders. So they seem to be clinically relevant. So the first thing we did is to have a look at expression of these molecules. And as I said, we carried this microarray where we dissected out these two streams, and as you can see quite nicely in here, this is a antibody staining on these GAT67 mice. And as you see, you can only see yellow staining in one of these streams, which sort of matches what we thought we would see from the microarray, that they're only expressing one of these streams and not the other one. When we look at the knockout mice of these, so this is towards the end of development, when all the interneurons should have got into the cortex by this stage. What we noticed in the cortex of these mice by carbidula staining is that we noticed a lot more interneurons in the cortex, quite similar to what we saw with the robo mice, which is a bit unusual. And again, similarly, when we looked in the cadherin 13 mice, again when we looked in the cortex, again we only see yellow staining in one of the two streams. And then with this mouse, when we looked in again using carbinin or HA6, which are two different neuron markers, looked at the end of development of the number of neurons in these mice. What we noticed was that actually with this one we see the opposite. We see a lot less interneurons in these mice. But the one thing we did notice when we look at the expression, when you look quite late on during development, there's a lot of red staining in this region, and it's just the interneurons and the pyramidal neurons seem to express this gene. When we looked a little bit further into this, what we notice is that because we see less interneurons in the cortex, it's not because there's a problem in either their generation or their migration, because we don't see a defect at E15. We only see it later on, because when they're already in the cortex. But what we know is that from other work with this gene is that if you knock this gene out, the cells are more sensitive to oxidative stress. So we think that when we look for apoptotic markers in these mice, we actually see more apoptosis, suggesting that the neuron's not surviving, which may hint that maybe this is one of the mechanisms why you see neurological defects in these mice. So conclusion of this part is that interneurons seem to express cadherins in specific streams at early stages, which is when we carry out a microarray study, and that loss of function, particularly cadherin 8, leads to increased number of cortical interneurons. So we were interested in whether there's actually a connection between cadherin 8 and robo. We know robo robo 1's been shown to interact with other cadherins, but we'd be interested to see how it interacts with cadherin 8. And also we noticed that cadherin 13 loss of function, it seems to lead to reduced number of interneurons at E18, but not an early stage. And this seems to be due to increased apoptosis. So it looks like some of the data we're getting looking in sort of developmental aspects in these mice sort of mimic sort of what you'd expect from the clinical data about the implication of these genes in neurological disorders. Although we would like to go away and try and do some behavioural studies with these mice and that sort of thing. So first of all, I feel like my collaborators at UCL, particularly John Palvez, who's like I've been working with for the last 10 years, and some of the other people that were involved in that, and also the people that provided the different knockout strains. Thank you.